have the pleasure of sharing with you some interesting work that we hope to be getting started on very quickly here to help overcome some of the regulatory and scientific challenges that have been facing us for some time. I will be presenting an evaluation of the safety and efficacy of an autologous adipose stromal vascular fraction cell product for knee osteoarthritis. And I'm told that after Dr. Veng's Ness, I have the second highest number of slides, so I apologize to those of you who had too much lunch today. These are my disclosures. I'm medical director for a company called Personalized Stem Cells, which is a sponsor for IND studies of GMP process stromal vascular fraction. I'm also co-founder with Dr. Malanga and Dr. Bowen for a registry called Data Biologics, and I'm also consultant for LipoGem. So these are all relevant to my talk today. Even though I'm a graduate of UCLA, I agree with my colleague from USC who states that stromal vascular fraction and ASCs are drugs in the language that was put forth by the FDA in 2017. And specifically, they are drugs because they are processed by exceeding minimal manipulation and may be used in a method that is considered to be non-homologous use. And those that do not meet these criteria are classified under the Public Health Safety Act as a Section 351 drug, and therefore they must be regulated as a drug. And just this week, as you heard, the FDA reaffirmed their position. The acting commissioner stated that we support sound scientific research and regulation of cell-based regenerative medicine. But at the same time, the FDA will continue to take action, such as issuing warning letters or initiating court cases against clinics that abuse patient trust or endanger their health. And so they're, as you heard, very interested in safety. And so they have as this week they were reaffirmed by a U.S. district court that ruled that the FDA does have the ability to regulate this space. Now that is for the adipose that is processed by enzymatic digestion. There are many different processing methods, and they are not equivalent. They're not equivalent in terms of their content, and they are not equivalent in terms of their clinical effect or biological effect. So I'll be focusing on the right there, the stromal vascular fraction. Adipose tissue is an interesting tissue. As you heard, is a heterogeneous collection of cells containing pericytes, endothelial progenitor cells, stem cells such as the adipose-derived stem cell or hematopoietic stem cell, and then mature cells such as the adipocytes, macrophages, other immune cells, endothelial cells. So we have this collection of cells. With the enzymatic digestion, you can isolate a subpopulation by removing the adipocyte, and there at the bottom you see a pellet that contains the adipose stem cell, pericytes, endothelial progenitors, and hematopoietic cells, among others. Now, if you take that stromal vascular fraction, you further culture it in the lab, and then pull off the plastic adherent cells and perform flow cytometry, you can then further characterize out different cells, and in this case, you can culture expand the adipose stem cell. You have a defined population of cells that's not quite 100% homogeneous, but almost. So you're saying, well, wait, at this point, I'm sitting here, I can't use these things. You're telling me that stromal vascular fraction and adipose stem cells are illegal. But the rest of the story is, unless they are studied under an FDA-approved clinical trial as an investigational new drug, not just an IRB study, but an actual FDA-approved clinical trial. So that is what we intend to do. So the FDA clinical trial, as you know, has phase 1 testing safety. Phase 2 and phase 3 evaluate efficacy. And once you have adequate scientific evidence, then you can obtain a biological license agreement, and this can become a FDA-approved drug. So what is GMP? Well, GMP refers to good manufacturing practice, which is a system for ensuring that products are consistently produced and controlled according to quality standards. Not necessarily the best product, but you have a consistent product that is safe and meets certain standard criteria. So these INDs must meet these GMP standards of safety, identity, strength, quality, and purity. So every drug you have in your medicine cabinet at home has met these criteria so they can be released to the public. GMP facilities are where these are produced, and we have such a facility in San Diego that we'll be using for creating a stromal vascular fraction. And these facilities are required to maintain and develop protocols and procedures for assuring quality control, manufacturing, consistency, laboratory testing, even how the products are labeled and packaged, et cetera. So why go through all this effort? Well, obviously, we want to see the field move forward. We want to develop well-characterized biologics that are safe. Furthermore, by characterizing the biologic and studying the clinical outcomes, we can identify those aspects that are providing improved outcomes, so we can advance our scientific understanding of these biologics. So currently, when I'm in my office, my patients ask me, which stem cell therapy is best, what works? Well, the first thing is we have a very long discussion of the current relevant literature, and then the second thing I tell them is that we don't do stem cell therapy. We inject multicellular tissue grafts, essentially. And it's always bothered me, which is better. We still don't know the answer, so we need to further study these biologics. So currently, these are the ones that we're used, and the outcomes have been good, but we need to move forward. We know that adipose is a rich source of MSCs, as you just heard. On a good day, on a good bone marrow harvest, I can get about 10,000 MSCs per ml, typically half that much. Whereas with the adipose, the same amount of adipose aspirated, 120 mLs, we can get 100,000 or more adipose stem cells per ml. If you believe that cell is the important variable for clinical outcomes, more might be better. I've always felt uncomfortable writing this prescription. Take unknown drug, unknown milligrams, unknown number of times daily, and that's exactly what we're doing currently with these multicellular tissue grafts. And despite the fact that we are seeing safety and we are seeing efficacy and we have some data to support our behavior, going forward, we're going to need to quantify. I just lost my monitor. So they're rebooting my monitor, so I'll look over here. So we do know that dose is important. One of our scientific advisor told me when I asked him, what is the optimal dose for a given medical condition? Well, the answer was more is better until it's not. And so there's an optimal dose for each medical condition, for each biologic. And this gives us the opportunity to study it. With stromal vascular fraction, by the way, we can get a greater number of cells. You can see we can produce up to 1 million adipose stem cells per ml. So we're volume reducing the adipose tissue by removing the adipocyte, and therefore we can inject into smaller tissues and pathologies. But the advantage is that we can actually count the cells with this protocol and know that prior to injection. Furthermore, we can characterize the biologic, so we can define what percentage of that product is adipose stem cell, what percentage might be pericyte or progenitor or hematopoietic stem cell. This will help us gain further insight into our biologics. Furthermore, we can test patient safety. We subject these tissues to sterility testing and endotoxin testing, and then because they don't contain adipose, there's no risk of fat embolism, as we have with the adipose grafts. And then once we have approval from the FDA to do so, we can store cells and culture expand them, and this allows for repeat dosing without having to repeat the harvest procedure. So I'm going to briefly run through some of the clinical trials. I'll mention that I have produced a summary document of the current published literature. You can obtain a copy at in the exhibit hall at our booth, or I can email that to you if you contact me afterwards. As was previously mentioned, there are some challenges to the current literature. Most studies are clinical case series with very few patients. Adjunct treatments are typically included, such as PRP or hyaluronic acid. They're often combined with some surgical procedure. The tissue analysis is typically retrospective after the procedure was completed. And most of these involve treatments that were where the tissue was produced at the bedside. And we have very limited number of prospective double-blind studies. PAC in 2011 was the first to use SVF in knee osteoarthritis and showed efficacy in two patients. Then in 2013, co-sampled SVF process from the infrapatellar fat pad and did second look arthroscopy and was able to demonstrate 87% of patients improved or maintained their cartilage status. And then the big study that was produced in 2015, the multi-center case control trial where 1,100 patients with severe knee osteoarthritis and other joints were evaluated with a single injection of stromal vascular fraction, on average 22 to 28 million cells, and showed that 89% of those patients had at least 50% improvement in their outcomes. And probably more importantly, even though 44% of those patients were surgical candidates, less than 1% actually went on to have joint replacement. Hong did this double-blind randomized clinical trial where patients served as their own control. They had bilateral knee arthritis. One knee was injected with SVF, and the other was injected with hyaluronic acid. And these patients were followed for 12 months. And if you look at the red line, that's the SVF group, and they showed significant reduction in their visual analog scale, and range of motion improved, whereas the control group did not show benefit. And this was also seen on the WOMAC pain and stiffness scores. And when they looked at the MRI a year later, the SVF group actually had some evidence of improvement on their Mocart and Worms score, where they look at cartilage repair. 69% of the SVF group showing some improvement on their MRI, whereas the control group, only about 6% showed improvement. And then just this year, Freitag produced a randomized clinical trial where patients received adipose stem cells. So these are culture expanded cells. To get those high numbers, 100 million cells, one group received one single injection. A second group received a booster shot at 6 months, and then there was a control group who received conventional conservative care. And in this study, we show that there was a significant improvement in both treatment arms and no change in the control group. And then I can't read this because I don't have my eyeglasses on, but essentially what this shows is that the MOACs, the MRI osteoarthritis score improved in the treatment group, and to no degree in the control group. Okay, so where do we go next? Well, VetStem was founded in 2002 and has been offering stromal vascular fraction to veterinarians and their patients for 17 years. Producing a GMP process, stromal vascular fraction, and adipose stem cells with more than 20,000 animals treated, including some exotic species. They are a preferred provider at San Diego Zoo, as well as the Wild Animal Park and SeaWorld. And who here would like to do liposuction on a northern white rhino? How cool would that be, right? So they also worked with the U.S. Navy Sea Lions and Dolphin program. So they have a lot of experience with animals, and now they've translated that experience to a company called Personalized Stem Cell that will sponsor IND and FDA-approved trials for human safety studies and eventually efficacy studies. And the benefit, again, is that we can measure cell count, cell viability, as well as cell identity. We have sterility testing and endotoxin testing. And this is a picture of some people working in the lab there. So it shows you what GMP processing means. You wear the bunny suit. You can't do this in your office. Don't try this at home. And essentially the flow is the patient has their fat FedEx to the GMP facility where it can be banked, and then doses can be produced and delivered back to the physician's office for treatment. And this is what the product looks like. So there's a quantified sterile product in that USP vial. Currently, we have a select group of physician investigators who will be helping us do these studies. We have strict FDA clinical protocols that they will need to follow. And our first study, we should receive our approval to start with by the end of this month to study stromal vascular fraction in unilateral knee arthritis. And this is a photograph of our current esteemed physician investigators and scientific advisors who will be helping us to do this work. And this is a look at our investigational new drug summary, where we're doing a phase 1/2A clinical trial on the safety and efficacy of stromal vascular fraction. Primary outcome will be safety and looking at adverse events, but also we'll have some preliminary look at efficacy, measuring KOOS and other outcomes. Anticipated further studies will include bilateral knees, multiple joints, and we are very ambitious. We have many clinical entities that we wish to explore. And I'm very excited, just like Flounder here from Animal House. Really excited to get this going. Thank you so much for your attention. I really appreciate it.